Hemoglobin, let's go for it. Right, everyone's favourite protein with quaternary structure that contains an ion. In fact, there's 4 ions per hemoglobin molecule. It's made of 4 polypeptides bonded together chemically. Anyway, underneath I put a little chemical equation, and we're going to fill this in first of all. So that the pretend chemical symbol for hemoglobin is Hb. It's not an official one, but it's there for shorthand. And if I have hemoglobin plus oxygen, I will obviously make HbO2. Okay, and then if I react that with another oxygen, I will make HbO4. And if I do that with again, I will make HbO6. And if I do it again, I will make HbO8. Note that these are reversible arrows, which means it can go this way and it can go the other way. In the lungs, when the blood passes to the lungs in mammals, I want it to go this way, so I'm making HbO8. And when it reaches my muscle cells that are working hard and using oxygen potentially, I want it to go the other way and release the 1, 2, 3, 4 molecules of oxygen. This has got a name, this molecule here. In fact, they all are by varying degrees, but a word that you should use in your answers is oxyhemoglobin. Oxygen reacts, or it binds it. Hemoglobin binds it, becomes oxyhemoglobin. Remember, technical terms get marks in exams. So we need to know how this works. And we're just going to go to a silly example first to kind of show you how it works for other things. If I had a room and I was heating the room up and I've got a metal object in that room, as the room gets hotter, so in a cold room, the metal object is going to be cold. As the room gets hotter, the temperature, it just heats up and heats up and heats up and heats up. There'll be a linear relationship between the room temperature and the metal object, okay, depending on how long the metal object has been in there. If I then replace, well, let's draw another graph this time with oxygen concentration, and then this is the how much oxygen is in red blood cell. Then you'd think you'd get, well, do you get a linear relationship? No, you don't. You get a line that's an S-shaped curve, and we need to know why do we get this S-shaped curve and what's the benefit of an S-shaped curve, because it provides an advantage. It's better that it works in this way than if it worked in a linear relationship. So you can forget about those now. So in effect, the reason why we have an S-shaped curve is because hemoglobin binds oxygen in different ways. The first one binds it's very hard for hemoglobin to do this first reaction. And this is very easy. So the more it binds, the easier it is to bind. So the first one's bonded really tightly. Well, when it binds the first one, that changes the protein structure that makes the next one more easily to bind, and so on and so on and so on. So if I drew a picture of hemoglobin, let's pop it at the top here. So it's a globular protein. So there's hemoglobin, and it binds 4 oxygen. 1, 2, 3, 4. But it binds them in varying degrees of affinity. So let's say that this one at the top is the first one it bonded. It's bonded really tightly, shown by the thick bond. The second one then binds more easily, but it's held less tightly. Next one's the same, and so on and so on. Okay, so this one's held the tightest, that one's held a bit slacker, that one's held a bit slacker, and this one's the most loosely held of all of them. So let's put this onto a graph. I'll do. And on the X is the partial pressure of oxygen. Which is similar to concentration. I was thinking, on the top of Mount Everest, there's less oxygen, but there's the same percentage oxygen as there is at sea level. So it's still 21% oxygen, but it's more spread out. So pressure affects oxygen concentration. If you squeeze a gas, the oxygen molecules get closer together. And I'm going to call this 12. 12, and the units that we do this in is the same as osmosis. It's kilopascals. That's the concentration or the pressure of oxygen in our alveoli. So this is in alveoli. So it's the concentration we'd find. On the y-axis is the percentage saturation of haemoglobin with oxygen. So the line is this S-shaped curve. So let's pop that on and let's go for purple. That'll do. So this is normal haemoglobin. So at 12 kilopascals, we'll go from here all the way up to the top. It's hard to draw these straight lines. And go across to here. Yeah, it's 100% saturated. Okay. And then let's pick a point here. Let's call this respiring tissue. So if this is a muscle cell doing work, so that's about 50. So this is about 30. So hemoglobin. So red blood cells contain trillions of molecules of hemoglobin. When they pass through the alveoli, they get 100% saturated. They then travel around the body. When they reach this area here, let's call that about 6 kilopascals. So this is some muscle tissue doing some work. It's using oxygen. When the blood cells reach there, the hemoglobin can only hold 30% oxygen. Therefore, it releases 70% of the oxygen it's carrying. Okay, that's not good enough because it's pointless red blood cells going past muscle that needs oxygen and then not giving it the oxygen. And therefore, we have the ability to change this curve. Yeah, that when the muscles are respiring, they make more CO2. CO2 dissolves and forms an acid. Around a muscle, you get a pH drop, and that affects the structure of the protein. And that means that instead of it looking like this, it still starts at the same place, it still ends at the same place, but it shifts it to the right. And this is called the Bohr shift. Yeah, and it's due to lower pH, i.e. more CO2. CO2, non-metal oxides dissolve and form acids, don't they, from GCSE chemistry? This now means that it still gets 100% saturated. However, when it gets to here, it's now, what's this value here? Well, that's down at 8. So 92% of the oxygen is now released to the tissue. So if it shifts to the right, we always write this. So shift right, or just is right in comparison. Yeah, and you can understand this, or you can just learn the answer. So because it's giving up more oxygen, the hemoglobin is held less tightly. It's like it wants to go this way rather than this way. Yeah, so these bonds have got smaller, all of them. So the affinity for oxygen is less. So hemoglobin has a lower affinity. That's the bit that people struggle with for oxygen. So now I can say at low partial pressures, of oxygen, hemoglobin releases more. Oxygen. So if I put oxygen twice in a sentence, therefore there's more oxygen to perform aerobic respiration. Happy days. So we have the Bohr shift, which means that we can give more oxygen to our tissues. Okay. The S-shaped curve, incidentally, means that it doesn't give up the oxygen very easily at the top either. So when it comes out of our lungs, it doesn't bother giving oxygen to other lung cells when there's loads of oxygen around. It kind of holds on to it for a bit. So that's a shift to the right. You could even have here, this could be bird hemoglobin as well, like a pigeon. So that's shifting that way. So let's shift it the other way. There we go. So this is now shifted this way. This is like fetal hemoglobin. So I have to just budge this up slightly. Budge it to there. That's our shift right. So that's the red one, isn't it? Good. That's the shift right. So yeah, shift left. Or just is left. It can shift left. If you hyperventilate, you lose your CO2 from your body, your pH goes higher, haemoglobin affinity, it holds onto it more tightly. People have to breathe into a paper bag to re-breathe in the CO2 because the blood cells are going around the body, just not releasing oxygen. So you get a perception of suffocating, which if anyone's hyperventilated, they'll know about. Anyway, here I just say kind of the opposite. So HB has a higher affinity for oxygen. Okay, so far. Now, instead of focusing at low partial pressures, well, sorry, we still focus at low partial pressures. So at lower partial pressures, hemoglobin can be more fully saturated. So it can take oxygen from the environment. This is what like worms can do. Therefore, there's a supply of oxygen for aerobic respiration. So I'm kind of saying the same thing here, aren't I? It's like, well, it seems bizarre that they both give the same advantage, and that's why people find it tricky. I find that it's well, what is the advantage? If it shifts to the right, you're a highly aerobic animal. You're a fast mover. If you shift to the left, you're a slow mover. You have to work at lower oxygen concentrations. So if we look back to this graph, and let's look at this, at this 6 value, the blue line now is it will only release a very small amount of oxygen. It's going to be about 60 here, so about 40% of the oxygen is going to be released. That means that the organism gets oxygen, but it doesn't get lots of oxygen, but it does get some. But it means that if you're a slow moving organism, you can get away with it. Okay, so the blue line, the example is a fetus. That it's got a higher affinity of oxygen than its mother, its hemoglobin does. Therefore, its hemoglobin can take oxygen from its mother's hemoglobin. Therefore, it has some oxygen for aerobic respiration. Admittedly, it can't, sprint up the stairs, but it doesn't need to because it's just sloshing around in a uterus, growing slowly, doing the occasional little punch and kick. Okay, so it doesn't need to. So if it shifts, if you're on the left, you're a slower organism. If you're on the right, you're a faster organism. Okay, we do have a source, a type of hemoglobin that its curve would look like this. Let's pick another colour. To go for green. So we've got one that's like that in our muscles called myoglobin. So that's myoglobin. So what would you say about myoglobin? Are we going to say the right answer or the left answer? Well, we'll say the left answer. Hemoglobin, in this case myoglobin, has an extremely high affinity for oxygen. At low partial pressures, it is fully saturated, which means it releases oxygen only in like desperation states. So you can see here that there it's fully saturated, and then it's got 0. So at low partial pressures, it releases all of it. If I drew myoglobin, it'll look like that. There's myoglobin, MB, and it just has one oxygen attached. It's got one heme group, and therefore, but it's bonded immensely tightly, and it's like our emergency oxygen supply in our muscles. We have 2 types of muscles. We have, well, in terms of colour, we have red muscles and white muscles. If you think of like a chicken, they have red meat on their legs, because they're running around all day, and they need a supply of oxygen inside those muscle cells. Whereas their breasts, that they use for flying, well, they don't fly anymore, and therefore they just use them very occasionally. Therefore, they don't need a supply of oxygen because they're only going to use them occasionally. They're okay doing anaerobic respiration from them. Whereas, a pigeon, its breasts are really red because they're full of myoglobin. So if you eat red meat, you're eating myoglobin laced cells, and if you eat white meat, then you are eating muscle cells that don't have myoglobin, the type of muscle cells that just do anaerobic respiration. If you don't eat meat, then you've got the moral high ground on us all. And on that point, I shall stop. Moit, new word. Write it down.